Para dar continuidade a essa programação, convidamos agora a professora doutora Cinara de Cássia Brandão e a professora doutora Lília Maria Garcia Bahia de Oliveira, que irão moderar a mesa redonda intitulada Uma Só Saúde na Toxoplasmose, experiências mundiais. Professoras, é a palavra de vocês. Mais uma vez, obrigada.
Obrigada, Fernanda. Nós vamos apresentar agora a mesa redonda de Só Saúde e eu vou apresentar as professoras em inglês, tá bom? Espero que todos aproveitem essa excelente oportunidade dessa mesa redonda. Isso é importantíssimo, complementando, né, a palestra que o Edilson deu, e essa mesa redonda vai ser muito importante para nós. So I would like to ask for enjoy our roundtable with Professor Pikka Jokelainen. We will have Dr. Laís Pardini as well, and Karen Shapiro. So I present you and thank you, Dr. Jokelainen, to accept our invitation. And it's a really good opportunity to have you talking to us in our event. And Laís, nice to see you again. It's a pleasure to have you here. Karen is just coming. She is here and just coming to our room. So I represent Dr. Jokelainen. She's the head of function for infection disease preparedness and One Health at Statens Serum Institute in Copenhagen, Denmark. Dr. Jokelainen has published about 130 scientific articles, mainly in the areas of One Health, zoonoses, infection disease preparedness, and science policy interface. They hold leading roles in major European One Health and preparedness initiatives. Thank you again to be here with us.
Thank you so much. Thank you so much for the invitation. Great to be part of this event, even though I'm here in Denmark, in Europe, so different time zone, but here I am. Yeah. I will share my screen, just a second. Comes here. And you should be seeing my first slide here. So indeed, my name is Pikka Jokelainen, and I'm really happy to start this session with sharing some experiences and also reflections and thoughts from a European One Health initiative focusing on Toxoplasma gondii called TOXOSOURCES. Let me go to my... Starting with this One Health approach for Toxoplasma gondii, I mean, it is a zoonotic parasite that has so complex life cycle and it presents everywhere globally. So it is really a One Health challenge. And it is also something that brings people together in a One Health way. I was browsing the program of this event too. I mean, so many different aspects are covered and discussed. So in a way, this parasite is bringing people together. From my background, I would like to mention that I am indeed head of function for infectious disease preparedness and One Health at the Public Health Institute, Statens Serum Institute here in Denmark. And then I have affiliations also at universities. I am very much biased towards collaboration. I think especially One Health topics, it's a wonderful thing to do together, and also we cannot do it without each other's expertise, and we all contribute with expertise, ideas, thoughts, roles into this. And especially I would like to highlight both these One Health, so cross-sectoral, cross-discipline, transdiscipline approaches, but also international approaches. So as mentioned, I have quite many roles in the European level, international initiatives, but also globally. And one key thing I would like to give a small promotion notice is that I'm currently the president of the World Federation of Parasitologists, and we organize this International Congress of Parasitology once in 4 years. And the next one is next year, 2026, in Canada, in Montreal. So I hope to see many of you there to really bring this expertise there is in your region on Toxoplasma to the global discussions. Well, Toxoplasma really is as a zoonotic parasite a One Health challenge. And one of the reflections I would like to bring is that when we work with or on this parasite from our point of views and focuses to try to see the bigger picture and how things are interconnected. For example, just looking at the life cycle or transmission illustrations, how things are actually linked. If you think about these 3 infective stages and how Toxoplasma infection can be acquired by many ways, and actually, indeed, most of the infections are acquired. So after birth, during the lifetime of the host, both humans and many animals. That is illustrated in this figure, for example, by those arrows that go to the adult humans. But if we think about the congenital infection, that is actually also in a way and further arrow from an acquired infection of the mother. So if we can do something about preventing infections, the acquired infections, that would be wonderful to do that better. To simplify those arrows going to the humans there, we can say like there are 2 ways. There is the meat-borne pathway, so eating the tissue cysts in meat or other tissues of infected animals, and the environmental pathway, so ingesting oocysts that can contaminate water or food or the environment. And practically this environmental pathway is a fecal-oral transmission, and these oocysts are shed by the infected felids in their feces. If you then think about the meat-borne pathway, so actually important to remember that the tissue cysts, they don't just appear there in these animals and their edible tissues or meat. Tissue cysts are there only in infected animals, and many of those infections are from the oocysts. So the environmental pathway is quite important in that field. And if we go further upstream the arrows, it's very important to remember that the oocysts do not simply appear in the feces of the cats. They can only be there if the cat is infected. So if we could stop that arrow, prevent the feline Toxoplasma infections better, then we would affect actually the whole picture. But focusing on the arrows going to the humans, the relative contributions of these pathways, what is the most important? What are the sources? Where does the infection come to us humans? That has been largely unknown. And it's clear, of course, looking at this image, for example, that there will be differences by setting, by region, what kind of food habits we have and how we are in connection with animals, environment, food, water. But really, this has not been investigated a lot for Toxoplasma gondii. And it's been partly, this kind of lack of source attribution has been partly because there's been not really good methods that fit parasitic infections. Similar studies have been done a lot for bacterial infections. And this is both laboratory methods and modeling approaches that have been lacking. And European Food Safety Agency, their scientific opinion from 2018 listed different approaches that could be used for Toxoplasma gondii source attribution. All with limitations, but also with potential. Of course, studies, case reports, outbreak investigations, European experience is that the possible outbreaks we have in Europe, we miss them. We don't even notice them. So not really investigated many of them. There's been a few quantitative microbiological risk assessment studies that have taken, say, like evidence on relative contribution of different sources. These have been mainly previously focusing on meats, different meat sources, different types of meats. Then it's, of course, a lot of discussion about serology. That would be brilliant if our serology approaches would be able to say if the infection is from oocysts or from the tissue forms. And this kind of methods used at population level would be really useful, but this is currently not yet in a major use. Genotyping, linked to the next talk, also can be relevant to really identify the same source and in the infections. But practically, at that point, 2018, and still now, largely the way to think where do we get the Toxoplasma gondii infections with humans, it's been based on experts' opinions. And that is a bias. That's one way, but that's quite biased and quite limited way. And the conclusion of this expert opinion from the European Food Safety Authority was that there is really a gap understanding which of these routes and what is their relative importance so that interventions could be targeted better. And this is an important gap and problem because Toxoplasma gondii is a priority pathogen both globally and also in Europe. This is the ranking lists for top priority foodborne parasites. And their Toxoplasma gondii is high both globally and also in Europe, even actually higher. So this was kind of acknowledged that we have a priority to look into this. And luckily, this need to know more about this and look into this was turned into a funding opportunity. And as a result, we got this TOXOSOURCES project. TOXOSOURCES is or was, because the formal time period of the project has already ended, but as a type of project is a joint research project under a larger umbrella, like a group of projects of One Health European joint program. And the TOXOSOURCES consortium, we were more than 20 partner institutes across Europe. Formally, we ran for 3 years and had a budget of 3 million euros, partly paid by EU, partly paid by the organizations who participated. And when this formal time period ended, then we still wanted to keep going and continue looking for funding and are still actively publishing, for example, the results and finalizing many of the analysis. I take us a little bit to the bigger picture because this is also an interesting experience, how our Toxoplasma project was part of this bigger, larger, like a program of projects. So this One Health European joint program, this was a very like a large One Health collaboration and initiative in Europe, started 2018 and went to September 2023 with a 90 million euro budget and 44 partners across Europe. And the partners of this one were all organizations that have some kind of mandate to work on topics that are relevant for One Health. So they actually had their like national ministries have said, you need to work with One Health. And this is how this setup was. Of course, that's a bias. We lack some expert organizations. At the same time, it was really capacity building with close links to national stakeholders. And the whole project worked really closely with European and global organizations, including the agencies in Europe, and then also the tripartite WHO, WOA, and FAO globally. So quite a unique setting to have a project in. Overall, this One Health EJP aim was to strengthen collaboration and improve preparedness, focusing on foodborne zoonoses. Our Toxoplasma project was under this one. Focus was also on antimicrobial resistance and emerging infectious threats. And all the work was along this different, like a strategic line that we want to improve laboratory methods, maybe improve surveillance data and so on to strengthen at European level preparedness for infectious diseases in a One Health way. And there were altogether 30 projects. So TOXOSOURCES was just at one little thing in this large soup of projects. But that was really interesting to be part of. And this is maybe also experience I would like to share, that sometimes let's join forces with others who work with something similar. Maybe they are looking at a virus or a bacteria, but maybe we have something in common and we could together make larger projects or initiatives across different topics. TOXOSOURCES as a project was, in its simplicity, this was our question. What are the relative contributions of different sources of Toxoplasma gondii infections? Mainly focusing on humans, but thinking of those arrows, actually also animals. So this is a picture from our hybrid start meeting in 2020, just before all the lockdowns hits our area. And how we approached this question was that we really all got to use our expertise and interest areas and what we can do on this, also what we like to do. That was really nice to see when people can bring their passion in it. So multidisciplinary international collaboration and different approaches, while indeed hit by a pandemic. So there's already, of course, most of the meetings were online meetings, and managing a large project during the top pandemic years was an extra challenge. At the same time, I would like to say that's also a reflection and experience. There was one PhD student who attended, who at the end of the project said, like, it was so nice experience to see how people who want to work together, how we can find a way, even if, like, you know, laboratories are closed and it was really hard time for everybody for different reasons, but somehow together we were able to do and continue, and this person got their PhD from these projects. Here is a list of the kind of work packages that the project was built on, and with their leaders and co-leaders. And generally, we really were building on previous work. What has been done before? How can we build stepwise on that? And we aim to take benefit of this, that it's quite rare to have more than 20 organizations working together on Toxoplasma. Like, what can we do together? We were thinking large reviews, multi-center studies, and so on from the beginning. And this is also like an I would say an experience and something to think about. Like, we in science very often go to a small thing, but if we can find a common question that we all can contribute to answering by different approaches, that's a way to bring people together. And I have just a selection of examples what in these different work packages we were achieving and doing. First one is this work package focusing on the multicenter quantitative microbiological risk assessment for Toxoplasma gondii infections. And this is an approach that has been done before for bacterial pathogens, especially for Toxoplasma, mainly focusing on meats. And outcome is quantitative estimate for some region or country is our unit in this. What are the main sources? And then interventions can be planned on that. A key part of this was to look into the literature, and we conducted large literature reviews. Here are figures of two of them. First one is a systematic review on prevalence of Toxoplasma gondii in different animals, including domestic animals, wildlife, and cats, phelids. And another one that was published just recently is a review on seroprevalence in humans and seeing how that goes in different regions, with the focus on quite a lot of focus on exactly on the geographical differences also within Europe. The image there shown is from the human seroprevalence article, and there are different regions. On the right there is the northern and the UK area where the line is very different, and this shows seroprevalence by age. So the x-axis is age in years up to 100 years, and y-axis is the seroprevalence up to 100%. And there's clearly a difference that in the northern part of Europe, it doesn't reach so high seroprevalence rates as in the southern parts. Another key part of this work was a quantitative exposure survey, a very large survey we did in 9 countries where there like a large population, like share of the population got a questionnaire to answer how they could be exposed to Toxoplasma, basically. So both what they eat, also gardening approaches, and many different questions that would capture the different exposures and where they could be, what could be the sources. And early results of this have been already interesting, how different diets and ways there are in different countries. And the final analysis of those data is still ongoing currently. But really nice to see like how different approaches, like what do we need, how can we compare across countries, can also help a lot in designing some interventions. Another focus was fresh produce, so vegetables, salads, all this, because indeed there's been a lot of focus on meat, but that's not the only way to be exposed to Toxoplasma gondii. And we here also we did some reviews. There's a couple of them shown, both like a generally just a review of how soil, water, fresh produce, and mollusk been contaminated, and then a specific review on the methods to actually detect this contamination in fresh produce. We focused on ready-to-eat salads because that's in Europe a foodstuff that is increasing in use. There's a lot of different options to buy in the supermarkets. And main point of this work was to do a very large multi-country study where we collected salads and tested them. And this was done again by step, like developing a standard operating procedure, which was implemented in many laboratories. There was a ring trial to see that we do it similarly. So the method was very well validated and then used in this study across countries. And the prevalence of oocyst contamination based on a molecular method, so we didn't identify like a viable oocyst with this method, it was 4.1%. Altogether more than 3,000 salad samples tested across Europe. So it is a proportion that is positive, and this is now then of course discussed because for this kind of salad types, focus is on bacterial testing from the industry side, and thinking about parasites is still quite new. Then one of the work packages was focusing on exploring serology for detecting Toxoplasma infections caused by oocysts. And this is very interesting. Like if we get this to work, this will be very, very interesting. Here, again, step-by-step approach, we first conducted a review to see what are the antigens described for this and how they work and what could be a good way to go more in depth to this. And main output from this is kind of like flow chart that is there shown on the upper part with the how to evaluate a new potential protein for this use. And then we did really the same work, starting again step by step, starting with bioinformatics selection of proteins of interest, including new ones and also those that have been already described in the literature, and then using well-characterized reference sera from experimental infections of pigs and sheep to see what it can say from there. And the conclusion from this work was that currently no antigen can be really giving a good estimate of the proportion of the oocyst-derived infections. But this approach is very interesting and the work continues. But I think it was also very nice to do this like step by step to see where if we want to continue this line, where do we put the effort in it, because this has potential. The final approach we used was focused on genotyping. And here the European challenge is that we mainly have the classical genotype number 2 here in Europe. And thus many of the typing approaches, they don't tell enough. You cannot say anything relevant based on those in our setting. So here we had two main outputs. Was that we were harmonizing the microsatellite typing approach that is used quite widely here across laboratories, and gave recommendations for how to optimize that and get more comparable results. And then we were developing also a new typing tool that is suitable for the European setting. And this new typing tool, again, step-by-step approach, I would say a key achievement was just the collection of the samples and isolates for whole genome sequencing. That really also showed how people want to work together and share their strains they have for contributing this work and really testing it with real samples in the European setting. And this is the summary of the experiences, is that, I mean, of course, there's the science. We really answered many questions. We're able to collate and compile new data sets that are really important for answering the research question. But this that we call cross-sectoral, we are different expertise areas, work across different countries, thinking about the One Health approach and collaborate. Somehow, this thing is the key take-home message. Like the feeling how to do it became even more interesting and relevant in that sense, that there's really a good drive to work further also together. So yeah, of course I'm biased, but I think it's a strength to have a cross-sectoral and multidisciplinary collaboration set up, and that gives, like, everybody's learning something new, because we all come to this kind of with our expertise areas, and then suddenly there is some new things we can learn together about. And that's all from my side. Thank you so much for your attention, and thank you, of course, to the consortium TOXOSOURCES.
Thank you, Pikka, for your presentation. Lilian, would you like to develop the question later, or can I get right now?
I think we can open for questions to the audience, because
Okay. So thank you again, Pikka. I read some notes. Joanna from Federal University of Fluminense is asking for you. The TOXOSOURCES project sought to quantify the relative contribution of different infection routes. What are the main findings regarding the balance between zoonosis-borne and meat-borne transmission, and how should this inform public health policies?
Thank you. This is really a good question, and this is also important. That how do we get impact of these results? I mean, the final results of really what are the, because it's per country we will have the results. What are the main sources? Then when we can say, like, okay, in this country is this type of meat product or this kind of vegetables that goes high on that list. Then there can be directly discussions about that, and maybe some advice given by the health authorities in that country based on the knowledge. At the general level, I think one of the key outputs is this, like, just thinking that there are many sources, and the focus on just on the meat as a source is kind of like only part of the picture. Taken especially, this was a major work, the big study, multi-country study on the ready-to-eat salads. And they are really salads that are meant by the consumers to eat without washing. And then if we there can with that was a molecular method, but can show Toxoplasma gondii contamination. So maybe those, like, the industry has been very interested, and we've been collaborating with them too. Like, maybe they want to improve their methods for that. And also this could be some public health advice given, like also rinse the salads that are sold, so that the package says you can eat it without rinsing, but maybe we should.
Thank you for your answer. I have a question. Again, congratulations, and thank you again for joining us today and share your experience. In your presentation, you discussed about the cooperation for knowledge exchange between countries and research on the topic of the One Health, Toxoplasmose, and other fields to be researchers. Right now, the Erexis, Latin America and Caribbean have been constantly publishing calls for cooperation. Considering this type of call, what are the opportunities or possibilities for cooperation with Europe and your group and other ones with South American countries and Brazil, Argentina, Colombia, and others? And just to show to our students that is now our audience, who can receive the benefits about that? Doctor, student, PIs, postdoc, graduation or undergraduate? How is that? Just for us to understand a little bit about that.
Thank you. Really, really good question. I mean, definitely, I'm just yes for all collaboration and finding the opportunities for that. Sharing experiences across different areas globally. It's so important for Toxoplasma gondii because we have so different settings, so different strains, so different systems. Like there was in the previous talk, like how are the systems? How do we recognize and notice these things? So we can learn from each other and share experiences and kind of build the capacity across. I'm very much also biased for the kind of the emerging, like the preparedness for when we think like we know how this is in our region, but what if it changes and what can we learn from other areas? Like the diversity in Toxoplasma strains in your region is so different than what we see here. And we are like, are we ready for that if that changes and so on? So I would say like from both sides, we are also, and we have been already discussing sometimes when we see some opportunities, should we aim for a common application and search? And it's very important that we have regionally networks that can quickly, then we can put these networks together and figure out what kind of consortium could fit a call. So I think that's one of the kind of the preparedness for an application is that we know each other and have learned a little bit what the others are doing. So then it's easy to reach out. And then for like what's like who can benefit? I mean, I think it's definitely for the doctoral students, PhD students, and so on. It's really great if you can give opportunities for joining because it opens really the world and you can learn much more than just from your local experts. But I would also highlight the lifelong learning. Like in this TOXOSOURCES, it was so nice to see there were people who have been nerding on their bioinformatics protein thing, and then suddenly they need to apply that to making an ELISA to test animal samples, and they learn much more about like how is that. So suddenly, like within our fields, also we all can learn something new. So I think, yeah, let's just continue studying this parasite together.
We never stop. Yeah, thank you so much. Just let me see if we have another question. Just a moment. In the chat, we didn't have any question anymore. Someone else wants to do some questions? I will have one after everyone finished the three of it because it's the early bit and Andres as well. So thank you again, Pikka. We hope to have an opportunity to keep working, to keep talking about this with you, about the cooperation and everything. So now I would like to introduce Dr. Laís. Hey, Laís, thank you again to be with us today.
Hello, everyone.
Yeah, and to enjoy our event. Dr. Laís Pardini is a veterinarian and professor and advisor at the Immunoparasitology Laboratory of the Faculty of Veterinary Science at Universidad Nacional de Plata. And Dr. Pardini conducts research in immunobiology of infection, biotypical genotypes of Toxoplasma gondii animals, as well the relationship between Toxoplasma strains and the host immune response. So feel free to share your presentation and thank you again to be with us.
Okay, thank you for the invitation.
Mm-hmm.
I am very happy to be here today.
Yeah.
Okay. Could you see okay?
Yes.
Okay. Well, I'm going to continue with the challenges for diagnosis, isolation, and genotyping in Argentina because different to Europe, we were more separately. We need a red, we need a network like in Brazil for working together. This is a challenge for us because in Argentina, very recently, in 2022, congenital toxoplasmosis was identified by the Ministry of Health as a notified event, but only for congenital toxoplasmosis, not for only symptoms like ocular toxoplasmosis or even in animals, only in humans. And we have 3 reference centers. If we think in vet, we think especially in LINPA, the laboratory where I work, and also INTA, because it's an important laboratory, more related with the community, especially with big animals, with cattle, ruminants especially. Then we have for humans reference centers, we have the ANLIS, the Instituto Malbrán, and the Toxoplasmosis Laboratory, which is in German hospital. It's a special area from a big hospital, a private hospital in Buenos Aires, and ANLIS is a reference center for public area and receive samples from the whole country. And if you don't know or if you not have clear how is Argentina, we are in the south of South America, and our country is very long and it's large. If you see, I live here in Buenos Aires province, but here in this area, very near to Uruguay. And if we want to send a sample for analyze to a reference center from San Salvador de Jujuy, which is in the north of the country, or from the south in Ushuaia, we need or plane or bus transport. But the distance from San Salvador de Jujuy to Buenos Aires is more than 1,400 kilometers, and from Ushuaia in the southwest is more than 3,000 kilometers. And it's very far for us, and it's difficult to, for example, send samples for isolation because of the conditions of the transport. When we think in the choices to diagnose toxoplasmosis by serology in humans, the most used are indirect fluorescent antibody tests, ELISA, modified agglutination test, and also indirect hemagglutination. And this is very used in a public hospital, especially in the province with more problems, with more economic problems, because it's the cost is the cheaper, and also chemiluminescence, but in the main or in cities or in the biggest cities. In Argentina, there are an Argentine consensus. The last one was had written in 2020. This doctor, Ricardo Durlach, was the main He was an infectologist, and he passed away last year. And he worked a lot to obtain this consensus because it's the only one in Argentina, and it was written between private, public doctors, infectologists, gynecologists, and also vets, because I work with them, and Dr. Cecilia Venturini, the director of our lab. And also in humans, toxoplasmosis congenita in Argentina is the most study manifestation of toxoplasmosis in our country. For animals, I'm pretty sure IFAT is the most used technique, but we have a limitation because of the conjugates. And ELISA test, the most common were import, and it is very high, the cost of them. It's not common the use of ELISA test. And for wild animals in our laboratory, a doctoral student, a PhD student, she's working to perform a modified agglutination test, because in Argentina there are agglutination tests, but these are developed for humans and not for animals. And when we study when we compare the test, we didn't obtain good results with animal samples. Then we have the option of direct diagnosis, but as I showed you, we have a very long distance, and to transport the samples refrigerated to the laboratories, because the equipment and the possibility of have mice to make the isolation is only possibly in the Laís German hospital or LIMPA. And the distance is very long. Which samples could we send, or could we receive? In some cases, German hospital receive especially placenta or cord blood, or maybe liquid. Okay, the most common is placenta and cord blood. And for us, for vets, maybe we can receive oocysts from fecal samples from cats, but it's very infrequent. And then we made some epidemiological studies, and we obtained brain, for example, from chickens, like in Brazil, and muscles, especially diaphragm, because in Argentina, in the slaughterhouses, it's common to, it's mandatory to do trichinella analysis, because it's an endemic parasite infection. And we can analyze diaphragm from slaughterhouses. In 1994 was the first isolation in a cooperation with JICA, the agency of Japan, because our laboratory was founded with a JICA project. Okay, the ideal situation is to obtain the isolate, but we need the mice, and this is a limitation for the majority of the laboratories. And then culture them in verocells. Other possibility is to make immunochemistry, especially, for example, in samples from, recently I received a sample from retina, where an ophthalmologist detect different and unusual manifestation of ocular toxoplasmosis. Okay, we are working on that. And the advantage of this is we can see not only the parasites, we can relate this with the lesions. And sometimes this is a good indicator. And then, sorry, because here it is in Spanish, but we can see the pictures. Here we think in which samples or in which situations is easier to obtain samples, or for what, which is the purpose. For example, we take samples from chickens because we use them as sentinel animals. Horse, we need to study more in Argentina in them because Argentina exports horse meat to other countries. It's not common to consume horse meat here, but it's an export product. Then in cats and in dogs, especially in dogs, it's a differential diagnosis with Neospora caninum, another protozoan parasite, but it's not a zoonosis parasite, but it's a differential diagnosis. In cats with clinical signs, but we need to know what's happened in the population, in the normal population, the common population, not only with clinical signs. In pigs, we know more because we have a risk factors study in some thesis. And then we know in small ruminants, we have recent cases in ovine production where abortion, outbreaks with abortion. And in goats in the north, we know is presence Toxoplasma, and it's important because it's a family production system, and it's important for these families, for these people, the farmers. Then, of course, we made PCR, conventional PCR, but it takes a little time and it takes cost, and also not all laboratories made that. Which is the situation for us. Of course, we should, when we decide to analyze a sample, if we think in the best situation, we want animals with clinical signs because it's easy to obtain the isolates. And of course, then to make the PCR and genotyping, but this is not a common situation. When we want to study that and know what happened in the epidemiology of the country, we obtain samples from slaughterhouses, especially outdoor, for example, in pigs, because in indoor pigs it's not common. Is only we receive samples if the vets don't have other results, positive results for other infections. And ocular or immunosuppressed human samples is very difficult to obtain because of the quantity or the sample and the quality. In immunosuppressed humans, We have a work because in the list, a PhD student is working with immunosuppressed children after transplant treatment. The sample is very small, the quantity is very small, and the quality of DNA is not best, and it's very difficult to detect. The PCR, yes, okay, we obtain positive result, but when we want to genotype the samples, it's not very easy. And at the LIMPA, we obtain more than 20 isolates from animals and 11 from congenital human toxoplasmosis, only from congenital human toxoplasmosis, because we collaborate with the Sherman Hospital. They receive, especially, as I said before, placenta and cord blood, and they use mice to obtain the isolates, but they couldn't conserve the isolates in viral cells, and then because of that, they sent the isolates to us for genotyping and to preserve the isolate. This happened since 2012. Is a number of years, but it's recent. And it depends, and it's necessary to have a good relation with the people who works in the hospital, German hospital and Laís, because we don't have a network like in Brazil. As Pikka say, here in South America, in Argentina, like in Brazil, we have a good variety of isolates. And the human isolates, as I said, was obtained in collaboration of German hospital, very different genotypes, a good variety of genotype, especially #14. This genotype, the same combination, was obtained in wallabies and also from chickens and also from dogs. A dog with an immunosuppressed virus distemper. When we think what happened with the people, with the community, we know it's more common to have higher seroprevalence when the water source, the source of water is not the best, for example, in rural or peri-urban areas. There have not many studies to study that. Only in Chascomús, is a city very near to La Plata. It takes 1 hour to go there or to come here. And then in the center of Buenos Aires province, in the city of Tandil, where they study factors associated with the prevalence of Toxoplasma also. And as I said, in the pediatric, the children's after transplant, also we could obtain 2 genotypes, but our PhD students have to study more than 80 samples. 80 children, not 80 samples, 80 children. Thank you for listening. This is our situation in Argentina. I hope we can work together as Cinara, because we have a work to do together. Thank you.
Thank you, Laís, for your presentation. The question will be, please send the answer in the chat if you could, just about the time. Do you understand? So I would like, did you understand? And you can answer later, just about the time.
Yes,
Okay.
it's okay. It's
Thank
okay.
you again to be here with us and to share your knowledge about that. So Lilian will introduce Karen, please.
Yes, it's my pleasure to welcome Karen Shapiro. Good morning for her, since it's morning in California now. Dr. Karen Shapiro is a veterinarian and professor of parasitology in the Department of Pathology, Microbiology, and Immunology at the University of California in Davis. Her research centers on transmission and protozoal parasites at the human-animal environment interface, with emphasis on waterborne transmission linked to sea flow of Toxoplasma gondii and One Health impacts on wildlife, food safety, and public health. Please, Karen. Thank you.
Thank you very much, Lilian. Let me start my presentation. All right, and I'll put it in presentation mode and just let me know maybe by chat or somebody if it looks good. Okay, Pikka, thank you. All right, wonderful to be here. I think that I attended this meeting 2 years ago. Yeah, Lilian, in 2023 in Brasilia. So it's nice to be back with the members of this group. So my talk will share a story from California. It will be a little bit more narrow in scope than the previous two speakers. We'll focus just on one species, on California sea otters. And let me begin with an introduction of the species, as it's relevant to understanding the rest of the talk. So sea otters in California are considered a threatened species. The map here on the right shows that they really did extend the population all the way from Mexico and up actually to the north through Alaska. And the current population is delineated here in orange, and it's still a fairly small population because the species was hunted almost to complete extinction back in the 1800s due to the fur trade. So currently the species is recovering, but it is not delisted. It is still marked as endangered because the population has never been able to reach this threshold of 3,000 animals for more than 3 years in a row. So this graph doesn't show you the population when it was almost completely gone. It was believed to be almost zero. It was found to still exist in a small area of California in the 1940s and has been increasing until about this time here in the late 1990s. And that's when biologists contacted veterinarians to try to understand why this population was not expanding. And they found out that infectious disease was one of the primary reasons the population was stalling its recovery. And it was around then in a study that was done between 1998 and 2001 when Toxoplasma was identified as an important cause of death in this species. I will just mention that the population has gone up since then, but it has currently been declining in the last few years again, which is again why it's still considered endangered. So I know you're all very familiar with the life cycle of Toxoplasma, and we've seen some One Health life cycle examples shown in the last talks. This life cycle is a little bit unique because it really focuses on this land to sea flow. So one of the questions that our groups often gets is, how do sea otters actually get infected with Toxoplasma? You're all very familiar with the cat, rodent, bird cycle and how humans can be infected both from oocysts and the tissue cysts in meat, like Pikka highlighted. What we have been focusing on is really what happens as they flow to the ocean after rainfall delivers oocysts to the near coast, where we believe they then get accumulated in the habitats where sea otters live, and particularly in the prey that they eat. This talk is not going to go into the mechanisms of transmission or of transport. I'll instead focus on virulent toxoplasmosis, but that work has been published, and I'm happy to answer any questions on that if there are any. So what happens when sea otters get infected? So typically we see kind of the classic toxoplasmosis that we see in other species. So this is a sea otter found stranded that has CNS disease, central nervous system, with tremors. You saw the whole body tremors, obtunded, unresponsive, but otherwise the animal is in pretty good body condition. It's not too thin. You usually don't see external signs of trauma. And an animal in this condition would almost always either die or be euthanized. There's very few success stories of treating these animals. Oops, sorry. Okay, so we do know that a lot of sea otters are infected. So this is a prevalence map showing the proportion of the population that's infected in different areas. And we also know that there's some hotspot areas along the coast where more sea otters are infected, particularly in Monterey Bay here, and in an area that we call Estero Bay. So these areas that are highlighted in red are areas where more than 60% of sea otters are infected, and this is based on an IFAT test, so presence of antibodies. So one of the main questions is, why do not all sea otters die? So why does only a small fraction of sea otters die, even though so many are infected? And as you all know, in this room, there's a lot of reasons that can be explained for host pathogen interactions, why the host sometimes dies and why it doesn't. And one of the factors that we're particularly interested in is this question of parasite strain. So the questions we designed to test is, are there some Toxoplasma strains that are associated with higher virulence in sea otters? So the next few slides will go through results that I actually shared with this community a couple of years ago when I last spoke, but I feel like this background is important to understanding what we then saw in the last couple of years that I haven't yet presented. So this study was done on about 150 sea otters, and we used two methods. So one of which you've already heard, the restriction fragment length polymorphism, where we can amplify a section of DNA and then cut it with restriction enzymes and look at a pattern to compare it with other genotypes. And then the second method is the multi-locus sequence typing, which is more expensive, but it also has a lot more resolution because we can look at the entire sequence at a particular locus and not just at whether or not some restriction sites exist or don't exist. So here are the results, and I broke it down because there's quite a lot to unpack here. I mentioned that we looked at 150 sea otters. The reason why this is 116 is that not in all sea otters were we able to get a complete genotype. And not in all sea otters did we have complete pathology where we were able to categorize an infection as Toxoplasma not implicated, so it was a completely incidental infection where the inflammation in the brain was either absent or very, very minimal. Whether Toxoplasma contributed to death, so another 18% of cases where the inflammation in the brain was quite moderate and even severe, but it wasn't the immediate cause of death. And then primary cause of death, those are the sea otters where Toxoplasma was identified to be the immediate cause of death because the inflammation was so severe. So in the animals so let me back up. So for the results for RFLP, restriction fragment length polymorphism, we were able to discriminate type X infections, type 2, and these others which were recombinants. So we see this purple and orange. Now, the MLST will start seeing more colors, and that's, again, because there's higher resolution that allows us to discriminate between these strains. So whereas the RFLP put all of these strange as 1, as type X. If we apply the MLST, we can see these different variants of X or the purple. And the same with the type 2. So we're able to see more different types of reds and oranges when we apply the MLST. The main takeaway point here is that in the animals that died from toxoplasmosis, we only had type X or X variant strains. And statistically speaking, animals that died from toxoplasma were 29 times more likely to be infected with type X. So this type X or X variant did seem to be more virulent than the type 2 infections. And I just want to draw your attention to this kind of lighter fuchsia color, this type X variant that was identified in 42% of the animals that died from toxoplasma, and tie that back to terrestrial studies we've done in felids. And this is a study that was led by Dr. Liz van Wormer, who I think some of you know. What this map does, it allows us to compare the strains that we found in sea otters, particularly this X variant, which is purple circles in sea otters, compared with the blue circles, which is sea otters infected with any other genotype, and compare those with the cats, which are depicted in diamonds. And so Liz did find the exact same X variant both in feral cats and one bobcat. And all of these cats are in watersheds that drain rainfall into Monterey Bay, which is where sea otters live. So the power of this approach is that it really allows us to be able to connect sources to the host, the sea otter, where the parasite is killing them. All right, so I presented some of this when I was there in Brazil 2 years ago, and we thought we understood everything there is to understand about toxoplasmosis in sea otters, but of course, science is fascinating, and it will always teach you new things, and things change. And so during the COVID era, so this is back in 2022, about 2 years ago, the pathologists were just coming back to work after not being able to be in the office and do necropsies for a couple of years because of the pandemic. And one of the animals they opened up had something that the pathologist has never seen before. And I'll tell you exactly what it is that she saw. Then she saw this in 3 more animals that she processed in the next couple of months. So 3 of those animals died in 2022, just as they were coming back to work. But one of them was actually put in the freezer in 2020. Again, because of the pandemic, they weren't able to necropsy them, to do an autopsy on an animal. 3 of the animals are from this high-risk area, Estero Bay, and 1 is from Monterey Bay. Both of these regions we know to be high risk for toxoplasmosis. So what they saw is a condition called steatitis. So this is severe inflammation and necrosis and edema of fat. And this was found all over the fat of the animal. So normal fat, so this is in the mesentery, the connective tissue connecting the intestines to the body wall. This is normal, so very smooth, pale, non-edematous fat, compared to what they saw in the steatitis cases, which is very inflamed, swollen, red, angry-looking fat tissues. And I'm showing this for the mesentery, but every single fat tissue that the pathologist looked at, including around the heart and the pancreas and subcutaneous fat, had the same appearance of steatitis. Very different. Toxoplasmosis was not even on the radar when the pathologists were first recognizing this. Then on histopathology, they started to suspect toxoplasmosis. So here's an image of normal fat in histo. This is an H&E stain. So you see the vacuoles, the fat vacuoles, and very thin membranes of that adipocyte cells. And then in the cases that I just showed you, very, very severe inflammation by all these infiltrating white blood cells. And then on higher magnification, the pathologists actually were able to observe bradyzoid cysts, immature. We just call them protozoal cysts at this time because we don't know yet that it's toxo, replicating right next to an adipocyte. And a really striking example here in panel C of these zoids multiplying intracellularly within this adipocyte cell right here. The next stage is immunohistochemistry for Toxoplasma gondii. We usually do this for toxo and another parasite called Sarcocystis neurona, which can also infect sea otters. And in this case, the Toxoplasma stain was positive, and lower magnification and then higher magnification of staining of the zoids, the tachyzoids in this case, among all these infiltrating white blood cells. So at that point, we got samples to genotype, and we performed the MLST approach. We actually only perform MLST at this point in our lab because it does provide higher resolution, and looked at 13 loci and identified something that initially, when I looked at the sequences, I thought was just a completely weird, bizarre, atypical genotype because it wasn't in our normal library of reference sequences where we usually have all these X and 2 reference strains. But when we started looking at ToxoDB, we realized that this is actually not new to the universe of toxo. It's a strain called cougar that was identified in the 1990s, but we've never seen it. So it looked completely new to us. And this story of the 4 sea otters that I just mentioned was published in 2023. So what is this strain? So the cougar strain, also called TGCGCA1, standing for Toxoplasma gondii, found in a cougar, a mountain lion in Canada, is considered a rare North American strain. And again, it was first isolated in the 1990s following a really large waterborne outbreak in Victoria, Canada. So here we see this beautiful One Health example again of the importance of Toxoplasma, both in wildlife, but also in humans. And this cougar strain has been shown to be highly virulent in mice. I will caution that the cougar itself was not directly linked to the human infections in Canada, and that's because there isn't, as you can imagine, it's not easy to obtain material like that from humans. My understanding is that some labs may have access to the strain that was implicated in the human outbreak, but it hasn't been published. Phylogenetically, cougar clusters in this clade D, which is the same clade where we see the other Toxoplasma infections in sea otters. So type 2 represented by ME49 and X represented here by RE type 12, but haplotype 12, but not that closely related. And again, when you look at the sequences, they're really quite strikingly different. This strain has never before been seen in sea otters or any other aquatic species. So to my knowledge, it's only been found in the cougars and feral pigs, which were found in California, but about 3 hours away from the coast. This is a work from Chun-Li Su. So one question that we have been interested in looking at is whether this cougar strain in sea otters is something new that never occurred before in the California ecosystem, the coastal ecosystem, or if we just missed it over the last 20, 30 years. And we do think it's new, and this graph is the reason for that. So the earlier study I showed you about all these type X and 2, so purple and orange samples in sea otters are from animals that we looked at between 1999 and about 2009. And that was because we had the funding to do this active surveillance. And then there was a break in our work due to lack of funding. And we had one case report in 2015 about an abortion case. So this was a really interesting case in a sea otter female who was chronically infected with toxo but aborted, and she had several miscarriages actually. So it's the first situation we know of a chronic toxo infection leading to abortion in a sea otter. And then the cases that I just presented, these 4 cases, was published in 2023. So this is from the 2020-2022 animals. And then we have continued to do surveillance since then and have continued to see cougar. So far, we have 13 animals where we found this infection. So we've only been seeing type X and cougar in the last few years. I want to just really quickly touch on one other new thing we're exploring in the lab, very similar in some ways to what Pikka had mentioned, for not just being able to tell if an animal is infected or not using serology, but actually being able to tell more about the pathogen. In this case, it's not an oocyst discriminating antibody, but rather a serotyping test. So this is an ELISA that uses peptides that are derived from different amino acids of certain antigenic coding region of the parasite. There's a lot packed in this single slide, but I know that I'm running out of time. So just quickly, what we've looked at is the pattern of immunoreactivity to different peptides in animals that are either infected with type X in purple, 2 in red, or X in yellow. So these are violin plots. And there's different variants of the same peptide. In this case, we have a GRA6 on the left and a GRA3 on the right. And the main point here is that we saw statistically significant reactivity profile for animals that are infected with type X versus 2 or cougar for the GRA6. And for animals infected with the cougar strain versus 2 and X for GRA3. So using this technique, we might be able to discriminate the type of infection, the type of strain infecting sea otters before the animals actually die. So the RFLP and the MLST approaches that I just shared, we need tissue. Even better would be an isolate, and so we need a chunk of brain. We can only do that on a dead animal, whereas this method could theoretically be applied on animals before they die. So that allows clinicians who are treating sea otters that are stranding to be able to tell whether or not a sea otter is infected with a virulent or a less virulent strain. That's the promise of this method. Okay, so to wrap up, we have found this strain that appears to be new in the California coastal ecosystem, cougar, associated with a severe and new presentation of steatitis in sea otters versus the encephalitis we have been seeing beforehand. And we're also exploring this ELISA as a new tool for pre-mortem diagnosis of virulent toxoplasma in sea otters. There's, of course, still a lot of questions that we're currently exploring. We still have never found this strain in any kind of cat. So we're starting to look now in domestic cats and wild felids. And we're also unsure why now. Why are we seeing this strain now? Does it have to do with climate events like heavy rainfall that we see in something called atmospheric rivers in California? We're not sure. So we're looking at that as well. And there's many, many people to thank. The group of animals that respond to the sea otters that die. So this is the California Department of Fish and Wildlife team. My graduate student, who is now a postdoc, Devin, did all the work that I presented today. And this is Melissa Miller, the head pathologist, who is leading that aspect of the work. And the cougar work is now published as well. So this paper just came out a month or two ago. So if you're interested in that updated on what we're seeing with cougar infections, both of those papers are now out. And I can't see any of the audience, so let me rearrange the screen. I'm happy to take any questions. Okay, I see some chat messages.
Thank you very much, Karen, for your excellent presentation. Very interesting. We are open for questions. But first we have the questions addressed to Laís. Sorry. Cinara, can you assist me with this, please?
Yes, sure. Andressa is asking to Dr. Pardini. Your work is truly inspiring, highlighting both the challenge of a diagnosis and genotyping in Argentina and the remarkable genetic diversity of Toxoplasma. Considering the difficulties posed by long distance to reference centers, the identification of more than 20 distinctly isolated animals and humans, and the impact of reinfection with non-colonial genotypes. What strategies do you see as most promising to centralize diagnostic capacity and strengthen One Health surveillance programs in Latin America. Oops.
Yes, it's difficult to ask that, Andresa, because I think we need more support from the government to make the network, because the effort is made by the groups. But I think we should put our effort in diagnose by serology, and don't use especially indirect agglutination tests. Maybe use more IFAT for human infections, for human serology. I think it's better for us to understand epidemiology, and especially in humans and in animals. I was listening Karen and Pikka also. We need to study what happened in the coast of Argentina, also with the cougar, because we have here puma concolor, and I think it's very similar to the cougar of Canada. Maybe the behavior of the animals, no, the felids. And we don't know what happened in Patagonia, for example. We have a PhD student at the lab, and she's working with wild animals, but with introduced animal species, the exotic, feral pigs and other Neogale bison also, but we don't know what happened with our species in the area, and what happened with the sylvatic cycle. We don't know what happened with that, and if that influence in the people, because I have a question for Karen. If you know, the cougar or sea otters are it's usual for people to eat that, because in Argentina, in the south, for example, camelids, or in the north, it's not the common along the country, but yes, in some areas, to eat, for example, meat from camelids or from javali, swines, no? I don't know what happened with sea otters, and if you have some information about that.
Yeah, sorry, let me make sure I understand the question. Is the question that whether people eat the sea otters?
Yes.
Yeah, so it's a good question. In California, I would say probably almost never, because they're protected species. The only population where this might be happening is by native Americans in Alaska, where the population is not endangered and they have permission to hunt marine mammals as part of their cultural identity and sustenance. That said, sea otters are not, from what I understand, as appealing to eat, because their fur is very valuable. That's why they're hunted to almost extinction. So they might be hunted for their fur, but they're very lean. They don't have much body fat compared with seals. So seals, I think, would be consumed more commonly by native communities because they taste better to people. That's my understanding from speaking with the groups that do the Arctic research.
Okay, thank you.
Yeah.
Would you like to comment something, Pikka, or?
Yeah, actually, I could add to this also, like in the TOXOSOURCES study, one of the parts of this exposure survey was a question if there's some local dishes or some like a food culture that what is eaten in some areas, because if we only think about like European level, or if I think from the countries where I know most about, I would miss something. So this is also why we should discuss with others. And I'm originally from Finland, and I can mention there we have The only wild felid is the Eurasian lynx. It's doesn't seem to be a good host to be a definitive host for toxo, but it's like a mixing the whole picture because it's a felid, it's wild, and actually sometimes they are catching if people's domestic cats run outside, so they could catch the cats, and then you suddenly have totally different arrows in your life cycle. And even though it's not usually eaten, but there are some hunters who would eat some parts of that animal if they hunt. It's protected, but there's a certain number of hunting permissions given for that. So it is really local. This is what is interesting with Toxoplasma, that it's so successful parasites in so different settings, and sometimes the questions we have in our area can be also relevant in other areas.
Million. Can I support you?
So we have some minutes for more questions, I think.
I think so.
Yeah, so we have some questions in the chat for Karen, for Laís, yet that you can just answer later if you want. Can copy and
Okay, I see one from Rafael for Pikka and me. What kind of expanded surveillance approach do you think could help us better track parasites across coastal ecosystems, including marine mammals, reptiles, and other aquatic species, given the challenge with continental surveillance? Pikka, do you want to go first?
Well, I mean, I'm really for what I call One Health surveillance or integrated surveillance. So it can be, we often, there's a lot we don't know, but then if we would kind of like get the puzzle pieces there. So if we don't know about wildlife, do we know something about domestic animals? How are the data from the humans, environmental samples, and so on? So the wildlife and the coastal areas are part of this this whole ecosystem and system where somehow I feel like we piece by piece can get the idea what's happening. And like you presented, Karen, very nicely, this like when you then find something in the marine environments, and then your questions comes like, where did this come from? Is it in domestic cats, this strain? And like they are linked to each other. So I think that is the starting to look into that, and this requires collaborations too, of course, because access to different types of samples and knowledge about the methods that can be used to different animal species and so on. There's many questions, but yeah, I don't know. How are you doing? What is the next step, Karen? You are living in this.
So number one, money. The challenge is money. Because we have so many people interested in this work, it's not the lack of interest of human body. It's the money to pay them and to do the research. So what we're doing now, the new study that I mentioned that's allowing us to continue surveillance, is we're very fortunate, given the difficult funding climate in the USA, that we do have an NIH grant to look at the flow of different strains from cats to sea otters. So we started looking at domestic cats, wild cats, stormwater, rivers, seawater, mussels, and the sea otters. And so to me, the big missing puzzle is, well, there's two for the cougar. First of all, what cat populations are shedding at? There must be a cat, like it's here in the landscape for the sea otters to be infected. And where is it? How is it moving? Like, where is it in the environment, and where is it concentrating? And so right now, mussels, so invertebrate, filter feeding invertebrates, seem to be the most useful tool to look for Toxoplasma in the marine ecosystem. The problem is that even when we can identify the DNA, we don't have enough to genotype. It's been a huge challenge. We're trying to design new genomics tools to enrich the genome of Toxoplasma so that we can then look at specific targets for genotyping. But the classic RFLP or MLST, and we've tried 18S metagenomics, metabarcoding, they're just not sensitive enough. So we are looking at new tools to try to get a better sense of what genomes, not just is a parasite there, but what genomic identity is it along the different routes from the land to the sea. But honestly, I don't know that we'll be successful. It's even with a grant to do the work, it's very difficult to find and characterize this parasite in the environment.
We have two questions from Lillian and one for Andressa. Lillian, you want to ask what?
Yeah, we are something pressed for time, so what I'm going to read because they can follow us.
Yeah, Lillian,
This question is, can you hear me? Yeah?
Yeah.
Yes.
Okay. So, Karen, this question is for you. You can see at the chat too. So your presentation reveals the emergence for coke strain in sea otters associated with new severe clinical presentation. What surveillance or research efforts are underway to understand the current prevalence, geographical spread for this virulent strain in both marine and terrestrial hosts in California and other regions?
Yeah, so this new grant that I mentioned, where we are able to look at felids, different types of cats, and the environmental surveillance is what we're doing right now. We very much hope that we can maintain the funding. We're worried that it will be canceled at any moment. But for now, we're continuing the work. So we have active surveillance in the hot risk zones that I showed, where the sea otters appear to be dying from cougar. Unfortunately, we don't have projects like this in any other region, mostly because of money. I mean, there's many groups are interested. I know there's a group in New Zealand that is working on surveillance as well. And I'm very remotely affiliated with that. But it's hard to get, you need a lot of money and people power to do this type of surveillance, and I think it's a huge limitation. The resources are hard to come by in different areas of the world.
Thank you, Karen. And so we would like to thank all of you speakers.
Andressa has a question for you.
Oh, sorry.
Andressa is asking to Dr. Shapiro. Your presentation was truly wonderful. Congratulations. We deeply admire your work here in Brazil and in just a few days. I will have the honor of visiting your lab in California, findings on the cougar strain. So the question is, what do you see as the most urgent research priority to advance both marine wildlife conservation and public health?
Oh, there's so many. Thank you, Andressa. I'm glad you're here. I think so. I talked a lot about the coastal surveillance, so I won't talk about that anymore. I think one of the most interesting to me that spans both wildlife conservation and human public health is actually the work that Pikka presented on trying to discriminate routes of exposure. I think this oocyst versus brotozoid exposure is such an important question because that's really the key to how to prevent infections. And yeah, I wish, to me, that is one of the big challenges to try to address, is the work to sure we have the tools to develop tools that might work differently for different animal species, for humans, maybe even in different geographical areas. We know in Brazil that there's a huge, what we think oocyst-borne infections that are probably recurrent over people's lifetime that might be very different from the situation in Finland or France or California. So it could be that we need different tools in different places to answer these questions. Yeah, I guess if there was one next grant I would really like to see funded, it would be on this topic. But there's so many, not enough time to talk about all the challenges and things we should be doing.
Okay, so let's wrap up the session. And thank you again for being here with us. It was very good from you, and your presentations were excellent. So we have some announcements. We would
Yeah.
like to thank all of you and invite everyone to check out the oral presentations on the Padlet link. Tag us on the social media and be back on time after lunch. And that's all.
Just
Cinara, do you have some? In
Yeah.
Portuguese, perhaps we have to say something.
Okay, just
Oh,
to,
yes.
to actually talk about the ICOPA in the next year.
Mhm.
So please share the information for us.
Yeah, that would be our nice next time to meet by latest. So I hope this session also like inspired for international collaborations and networking. So there are many international events we can continue these discussions, and ICOPA for sure can be one of them.
Thank you so much. I have finished. Thank you again to be here with us, and I will say some advice to the audience. Thank you so much again.